Welcome back to AllAboutRx. Today, we're diving into the absolute masterclass your body puts on every time you eat a piece of avocado or a slice of pizza. We are breaking down the digestion, absorption, secretion, and utilization of dietary lipids. We're going to trace exactly how these fats get processed right from the start by acid-stable lipases in your stomach, emulsified by bile salts, and then completely dismantled by pancreatic enzymes. After that, we'll see how they manage to cross the gut barrier, get repackaged into these massive transport vessels called chylomicrons, and finally get burned for energy or stored by our tissues. Let's get right into it. Here's our roadmap for this explainer. 1. Gastric processing and emulsification. 2. Hydrolysis by pancreatic enzymes. 3. Absorption and mixed micelles. 4. Resynthesis and chylomicron secretion. And 5. Chylomicron metabolism and utilization. Starting off with part 1, gastric processing and emulsification. There's a really fascinating contrast between the highly specific limited digestion that happens early on versus the heavy lifting that comes later. In your stomach, you've got these acid-stable lingual and gastric lipases trying to work in a brutally tough environment. We're talking a pH of 4 to 6. And they have a super specific target, short and medium-chain fatty acids, basically anything with fewer than 12 carbons. But the vast majority of the complex fats we eat actually bypass this early step entirely. They just wait for the massive pancreatic processing that happens later on in the intestine. So you might be wondering, why do we even have these specialized gastric lipases if they skip the main fats? Well, they are absolutely vital for neonates. For babies, milk fat is their major calorie source, and milk just happens to be incredibly rich in these short and medium chains. This mechanism is also a literal lifeline for patients with cystic fibrosis. Because they suffer from pancreatic insufficiency, meaning they lack those later pancreatic enzymes, their bodies rely heavily on this early gastric processing just to absorb basic fats. Now, as the bulk of our complex lipids leave the stomach and drop into the small intestine, specifically the duodenum, they run into a classic problem. Oil and water don't mix. The duodenum solves this through emulsification. By combining the detergent action of conjugated bile salts with the mechanical churning of peristalsis, your gut essentially acts like dish soap on a greasy frying pan. It blasts those large fat globs into tiny, tiny droplets, which massively increases the surface area for enzymes to actually get to work. Which brings us right to part 2, hydrolysis by pancreatic enzymes. During hydrolysis, an enzyme called pancreatic lipase relies on a helper protein called colipase. Think of colipase as an anchor that holds the lipase right at that lipid-water interface. Together, they strip away fatty acids from carbons 1 and 3 of the triacylglycerols, leaving us with 2-monoacerglycerol. And working right alongside them, you have cholesterol esterase breaking down cholesterol esters, and phospholipase A2 systematically degrading the phospholipids. It's a total demolition crew. But these powerful enzymes aren't just blindly dumped into the gut. Oh no, this degradation is strictly, beautifully controlled by hormones. The moment stomach contents hit the intestine, a hormone called cholecystokinin, or CCK, is triggered. It tells the gallbladder to release bile and signals the pancreas to release its enzymes, while actually slowing down your gastric motility. Simultaneously, because the incoming food is highly acidic, a second hormone called secretin gets triggered by that low pH. Secretin signals the pancreas to flush the area with a bicarbonate-rich fluid to neutralize all that acid. To help lock this into your memory for test day, just think of the phrase, CCK calls, secret ensues, right? CCK calls for the bile and enzymes to get to work, while secretin soothes that highly acidic chyme with a neutralizing bicarbonate wash. Moving on to part 3, absorption and mixed micelles. At this stage, the demolition is done, and we're left with primary digestion products like free fatty acids and 2-monoacylglycerols. But remember, the lumen of the gut is watery, so these lipids combine with bile salts and fat-soluble vitamins, that's vitamins A, D, E, and K, to form what we call mixed micelles. These micelles are completely soluble in water. They essentially act like highly efficient little ferry boats, safely transporting the lipids across the gut's unstirred water layer and dropping them off right at the brush border of the enterocytes for absorption. Let's look at part 4, resynthesis and chylomicron secretion. Okay, what happens next inside the cell is absolutely fascinating. The smooth ER takes those fragmented, absorbed lipids and runs the disassembly line in reverse. It uses enzymes to resynthesize them back into complex triacylglycerols and cholesterol esters. But wait, now these newly formed molecules are super hydrophobic again, so the cell packages them into a core, coats them with phospholipids and a stabilizing protein called apolipoprotein B48 and forms a massive particle known as a chylomicron. This giant chylomicron is then secreted via exocytosis directly into the lacteals, our lymphatic capillaries, which will eventually dump them into the bloodstream. But actually, scratch that for a second. Remember those short and medium chain fatty acids from the very beginning of the explainer? Here is the one major exception to the rule. Because their carbon chains are shorter, they are actually water-soluble enough to skip this whole complicated micelle and chylomicron packaging process entirely. They bypass the smooth ER and just head straight out of the cell directly into the capillary blood. Pretty neat, right? All right. Last section, part 5, chylomicron metabolism and utilization. Reaching their final destination, the chylomicrons leave the lymphatic system via the thoracic duct and enter systemic blood circulation. As they squeeze through the tiny capillaries of your skeletal muscle, cardiac muscle, and adipose tissue, they encounter an enzyme stationed on the endothelial cell walls called lipoprotein lipase, or LPL. This enzyme hydrolyzes the chylomicron triacylglycerols right there in the blood vessels, releasing free fatty acids. Muscle tissue snatches up these fatty acids and immediately oxidizes them to create ATP for energy, while adipose tissue grabs them, reesterifies them, and stores them as fat for later. Once LPL has stripped away most of the triacylglycerols, you're left with this deflated, cholesterol-rich particle called a chylomicron remnant. The liver acts as our final cleanup crew here. It recognizes specific receptors on these remnants, pulls them out of the blood via receptor-mediated endocytosis, and breaks them down internally. That way, their cholesterol, amino acids, and glycerol can be efficiently recycled by the body. Nothing goes to waste. Now, it's absolutely crucial to understand how delicately balanced this entire system is by looking at its clinical impacts. For example, the anti-obesity drug orlistat intentionally disrupts this pathway by inhibiting those gastric and pancreatic lipases. No lipases, no fat absorption, which promotes weight loss. Conversely, if this pathway is pathologically disrupted at any stage, it leads to a condition called steatorrhea. This causes excess fat in the stool and, really dangerously, a secondary loss of essential fatty acids and those fat-soluble vitamins we talked about earlier. Before we wrap up, let's do a super quick recall of this incredible biological journey. We started with the acidic gastric processing of short chains. Then, duodenal emulsification increased our lipid surface area. Pancreatic lipases and hormones stepped in to create 2 monoisoglycerols. Mixed micelles ferried those products across the brush border. Enterocytes rebuilt them and packaged them into chylomicrons. And finally, lipoprotein lipase hydrolyzed them in our capillary so our muscles and adipose tissue could actually use the energy. It's truly mind-blowing to realize that a process as seemingly simple as digesting a single drop of olive oil requires this meticulously coordinated dance of specialized hormones, detergent-like bile acids, and cellular ferryboats acting precisely in sync. What other highly complex biological factories are running perfectly right under our noses? Thank you for tuning in to this explainer. If you found this breakdown helpful, please make sure to like, subscribe, share with your friends, and drop a comment below with any questions or topics you want covered next. See you in the next explainer.